All right. Let's begin. We're now going to be looking at Chapter 3. Chapter 3, dealing with what are referred to as macromolecules. Here you see it. Macro, meaning large, huge, stupidly huge molecules. Why do we call them macromolecules? Because these molecules are going to be so large, they're going to be composed of anywhere from thousands to billions with a B atoms. Now, for all living things here on planet Earth, you're going to see that there are four classes of major classes of macromolecules. The carbohydrates, which we call sugars. Lipids, better known as fats. And yes, you have fats all over, not just the adipose. But we'll see later in this chapter that lipids, fats are everywhere. Proteins. Proteins do the work of the cells. Proteins maintain the structure, the shape of the cells. Proteins are responsible for being the enzymes that regulate all metabolic functions in our body. And then the nucleic acids, the DNA, RNA, the means of saving the instructions. What instructions? The instructions on how to make and maintain you. Your dog at home, your cat, the tree outside, the bacteria living in your gut. All use nucleic acids. Now, as I said in one of the last lectures for Chapter 2, everything here is going to be organic molecules, meaning they are all comprised mainly of, centered around carbon. Now, to help us as we are looking at the making and the maintaining and what these are, these macromolecules, let's get some nomenclature out of the way. Monomers and polymers. Mono, singular. Poly, many. The mers, well, subunits. I like to tell students, if we were in lecture actually sitting there, you looking at me kind of all slack-jawed, laughing at my horrendous jokes. When you think monomers and polymers, think Legos. An individual Lego brick doesn't do much. It's a monomer. But you take a bunch of Lego bricks and you put them together as a polymer, well, a house, a plane, a spaceship, whatever. The example from your textbook. 2 monomers, 2 sugars, glucose, glucose. You add the 2 monomers, the 2 subunits, the 2 Legos, the 2 sugar molecules together, you get what is referred to as a disaccharide. Di meaning 2, saccharide, sugar. So 2 sugars bound together. And they're bound through a covalent bond. Now remember I told you last chapter to make covalent bonds, the sharing of electrons. Here the sharing of electrons is across that oxygen bond. It takes energy to make, thus it takes energy to break. You're going to need an enzyme. They refer to the making of this bond as a dehydration reaction. Dehydration reaction deals with water, H2O, 2 hydrogens, 1 oxygen, 1 hydrogen, 2 hydrogen, and there's our water. Notice they disappear. The oxygen over here now forms the bond, and we've lost, and there we go. Actually, their thing is wrong. Look at that. Oh my gosh. We'll have to call. So, here's the hydrogens and here's the oxygens. The textbooks are wrong. Yeah, authors, the artists are human people. So, Hydroxyl, hydroxyl. Remember our functional groups, the OH and an H. Two H's and an O, water. There's our water right there. Leaves us an oxygen and this open bond right there. So you can now have this special bond between the two sugars forming the disaccharide. Dehydration. Remember, to dehydrate something is to take water out of it. You dehydrate meat, make jerky, things like that. Well, if you want to break that special bond, you add the water back. And they refer to this as hydrolysis. It is the reverse of the dehydration reaction. So what you do is you add the water back. An enzyme will break that bond, or break, not that bond. It will break this bond. H2O, hydrogen and a hydroxyl. The hydroxyl goes back, and so does the hydrogen. To break it takes enzymes, because you're making and breaking covalent bonds. That just doesn't happen willy-nilly on its own. I mean, it can, but the time frame is measured in years and decades, whereas biological systems need reactions like that to happen in seconds, milliseconds. So enzymes are going to speed up hydrolysis and dehydration. If you were to take a disaccharide, sucrose, fructose and glucose joined together, same oxygen bond, you would take a bunch of that, dump it in water, and wait. Yeah, it would split, it would break, it would happen on its own. Days, weeks, months, years from now. But you take that same sucrose, put it into water, and you add an enzyme, sucrase. Sucrase, that enzyme goes through and cuts, does the hydrolysis reaction in seconds, milliseconds. Anytime you see a word that ends in ase, sucrase, which I just got done talking about, lipase, peptidase, anything that ends in ase means it's an enzyme. What prefaces ase tells you its reactant, what it works on, its substrate. Lactase, maltase. Lactase works on lactose, breaks that sugar down. Lipases go through and break down, cut, carbon by carbon by carbon, lipids.